Welcome back, students. Today we are going to discuss Unit 5, and in this particular unit, our topic of discussion is transport in plants. Okay, transport in plants, part 1. So, before I start the main content, I would like to inform that the images used in our slides are for education purpose only. We respect intellectual property right of the original creator and owner of these images. If you are the owner of any of the images used in this PPT, have any concern, any issue, please contact us at sathee@iitk.ac.in and we shall remove the images. In this particular video lecture, we are going to discuss these points: transport of solute and water in plants, transpiration and exchange of gases, stomatal mechanism, osmoregulation in plants, water relation in plant cells, and water potential. Okay. So, as you know, that plant doesn't have any circulatory system just like animals. Plant doesn't have. But they also need water. They need sunlight, right, to synthesize their own food and also to transport that synthesized food to other parts of the plant body. And also, movement of water is required from root to other parts of the body. Okay, so they need to transport water, mineral, nutrients, and organic nutrients, and they doesn't have circulatory system. Okay, but they need to transport these things from root to top of the water, and also they need to distribute the food. So for that, different type of movement of nutrients and water occur through different type of mechanism in plants. First type movement comes under this short distance category. Right. Short distance means movement of water and nutrient within cell or across membrane means आसपास की cells and from cell to cell. Okay, so what are the methods involved in this short distance transport? Like diffusion, like cytoplasmic streaming, and active transport. These are methods which are actually used up by the plants for the movement of any nutrient or water for short distance. But if long distance movement is required, that time plant require a specific system, means vascular system, which contains xylem and phloem. Movement of water occur through xylem and movement of food occur through phloem, right? And this movement of water and phloem, this process is known as translocation. Okay, as plant doesn't have any kind of circulatory system, but they also require to move food to all the parts of the body for its metabolic activity. They also require to move water from the soil to leaves, right, to synthesize their food. So two type of uh, mechanism are here. For short distance, they require different small processes like diffusion, cytoplasmic streaming, and active transport. And for the long distance transportation, they require specific tissue, means xylem and phloem, and this process is known as translocation. Okay, clear? Now, so following mechanism are used in plant for transportation. First, as just now we have discussed, diffusion for short distance movement. Cytoplasmic is also required for short distance and similarly active transport, and this vascular system for long distance. So one by one we will discuss all the type of methods. First is diffusion. As you know, diffusion is a passive movement of water and in which there is no energy expenditure occur, means movement occur from high concentration to low concentration, right? So molecule actually randomly move from high concentration to low concentration, as you can see in this particular picture. Okay. Eh, as you can see this particular picture, this here, this is the outsider membrane, right? And there is no molecule, right? So slowly movement of these molecule occur from high concentration. Here concentration is high, and here there is no concentration is low. So till then, when concentration on both the side of membrane, outside and inside, get equilibrium, it make equilibrium, or concentration is in equal amount, so it require no energy, nothing, just randomly any kind of molecule move from high concentration to low concentration. Okay. And within cell, between cell, and over short distance, this diffusion method is used up by the plant cell, and this is also critical for gaseous movement in the plant. So what are the factors which affect the rate of movement or diffusion rate? This is concentration gradient, membrane permeability, sometime temperature, and pH pressure also. Okay. So these are the factors. Now, next term which is related to diffusion is facilitated diffusion. Facilitated diffusion means this diffusion require a kind of molecule which actually facilitate the movement of particular molecule. Okay, this is again a passive type of movement, right, which require pre-existing concentration gradient. It require pre-existing concentration gradient. In the last previous slide we have seen there is no concentration gradient on one side, just outside the cell there is a concentration of a water molecule, there is no water molecule is present inside the membrane, so that doesn't require a concentration gradient. But for facilitated diffusion, it require a pre-existing concentration gradient. Clear? So this facilitated diffusion utilize a specific type of membrane protein which actually help in the movement of that particular molecule. So this facilitated diffusion require or utilize membrane protein to help hydrophilic substance cross the membrane. Hydrophilic substance, only hydrophilic, okay, to cross the membrane. These are highly specific, allowing selective uptake of substance. Highly selective, and it require selective uptake of substance. As you can see, this is a membrane. This is cytoplasmic membrane. Okay, plasma membrane. It contains certain kind of protein which is known as carrier protein. Okay, and this carrier protein is actually facilitate the movement of these insoluble molecule from outer side of the cell to inside of the membrane. It facilitates this diffusion. Okay, so certain kind of carrier molecule is involved. That's why this process is known as facilitated diffusion. Okay, and these protein channels, which is also known as carrier protein, here it is mentioned carrier protein, these protein channels form large pore, okay, in outer membrane of plastids, outer membrane of mitochondria, and in some bacteria as porine. Okay, it allow movement up to the size of a small protein to pass through. Okay, it allow molecule up to the size of protein to pass through. Like aquaproteins, these aquaporins, sorry, not protein, aquaporine, these aquaporine or channel protein, these are composed of some protein forming water channels. Okay, protein form a water channel, because normally water movement of water is also sometimes this aquaporine is required for the movement, so that's why it form water channels. Specifically, these aquaporine specifically facilitate water transport across the membrane. Okay, it facilitate water transport across the membrane. So transportation rate in this facilitated transportation reach a maximum when all protein transporter are used. There are so many these type of channels or protein transport occur throughout the membrane. So transport rate reach maximum when all the protein transporter are used. Okay. Now type of facilitated diffusion. There are three type of facilitated diffusion. First one is symport. This is symport where movement of a molecule occur in both the in any two type of molecule, means this is red one or black one, right? Both molecule move in the same direction, in the same direction. Okay, this is known as symport, same direction movement of one or more than one molecule occur. Antiport means two molecules. There are also two molecules, but movement of two different molecule occur in different direction. Means if this navy blue color molecule is able to come inside the cell, then simultaneously this triangle molecule will move from opposite direction, means from inside to outside the membrane. So we can learn that antiport movement in opposite direction. This movement occur in opposite direction. Same direction movement, symport, opposite direction movement, antiport. And third term is uniport, in which only a single type of molecule can move independently in one direction, or we can say in any direction, but in any direction, either inside or outside. Okay, but molecule is only single type of molecule. So facilitated diffusion are of three type: same kind of movement, same direction of movement, same direction movement, symport, anti-direction movement, antiport, any one molecule, single molecule movement in any direction, either inside or outside, but it move independently. That time we can use the term uniport for the same. Clear? Okay. Now, next type, all we have discussed diffusion, facilitated diffusion, now active transport. And these are the method for small distance movement. This method is used up by the plant for the small distance movement. So third type is active transport. If word active is present over here, then obviously we can understand that ATP is used up in this particular transportation. So this one is energy dependent transport which require ATP. Clear? Means it pumps move molecule against the concentration gradient. Means if this is a cell, and this is a cell membrane, concentration is low of concentration of any molecule is low outside the membrane, and concentration of any molecule is present in high amount inside the cell, but that molecule is required for that particular cell. So movement of this molecule against the concentration, means from low concentration to high concentration, and that time it require energy, because automatical movement occur only through concentration gradient, means from high concentration to low concentration through diffusion or through facilitated diffusion, right? But if the movement of a molecule occur from low concentration, as you can see, these uh, green color molecule are present in low amount in outside the cell, while this one is present in high concentration inside the cell. So movement occur from low concentration to high concentration. That time it require energy, it require ATP for pushing up the molecule inside the cell. So this is also known as uphill transportation. As we move upside, और अगर हम कोई वी कैन से अगर हम कोई पहाड़ चढ़ते हैं तो हमें ज़्यादा एनर्जी लगती है, ठीक है, जबकि पहाड़ से उतरने में हमें नहीं लगेगी। हम सीढ़ियाँ चढ़ेंगे, हमें ज़्यादा एनर्जी लगेगी, हम सीढ़ियों से नीचे उतरेंगे, राइट? हमें नहीं लगेगी उतनी एनर्जी। दैट्स व्हाई दिस प्रोसेस against the concentration, it is known as uphill transportation. Clear? So this is active transport, in which also some membrane protein is involved. This membrane protein, or we can say pump, carry that particular substance across the membrane, as over here this blue one. Okay, this membrane protein or certain kind of membrane pumps follow a method through which it actually carry that particular molecule and pump that molecule inside the cell. These are again very specific, sensitive to inhibitors also. So in this, transportation again reach a maximum level when all the transporters are used up. Clear? Now, comparison of different transport process. As you know, this is simple transport. Simple transport require any membrane protein? No, just concentration gradient. Concentration, it doesn't require any kind of protein. Is it these are highly selective? No. Is any transport saturation occur or saturates? No. Uphill, this one simple transportation is a uphill transportation? No, because according to concentration gradient, high concentration to low concentration, so it doesn't require any kind of ATP molecule or uphill transportation system, right? While in facilitated diffusion requires specific protein for the movement of molecule. These are highly selective and it transport saturates also. But this is again doesn't require any type of ATP molecule. So this is not a uphill transportation. Clear? While this active transport requires certain kind of protein, these are highly selective, transport saturates, and this one requires certain kind of ATP or energy to pushing up that molecule from low concentration to high concentration. That's why we can use the term uphill transport for the same. So this is a simple chart which can through which you can just compare all three processes: simple diffusion, facilitated, and active transport, which is involving short distance transportation of molecules. Now let's discuss some important points which is related to plant water relation. So as you know, plant water relation is essential for all physiological activities in plants and living organisms, as this water act as a medium for dissolving substance and suspending particle within the protoplast or not protoplast, protoplasm of the cell. As you know that woody part of the plants has less water, right? While the soft part of a plant has more. Some seeds contain water to remain alive and respiring. So high water demand makes water a limiting factor for plant growth and productivity in agriculture and natural environment. Okay, high water demand make water a limiting factor for plant growth and productivity in agriculture and natural environment. As you know that terrestrial plants absorb large amount of water daily, as most of the water is lost through transpiration. Still they need lots of amount of water daily. As if we can discuss over here some example like a mature corn plant. This mature corn plant absorb nearly 3 liter water daily. 3 liter water. While a mustard plants, this one, absorb its own height in water in about 5 hours. So that much amount of water is required by the terrestrial plant. Now let's discuss some terms which is related to this water and movement movement of water, movement of solute, movement or solvent related transportation. So first term which we are going to discuss over here is water potential. What is water potential, and what are those factors which actually determine its effect? So water potential is determined by two components. First is solute potential, which is known as psi s, and pressure potential with psi p. Okay, so this water potential is determined by two component, solute potential and pressure potential. Water potential is a sum of solute potential and pressure potential. So psi w is equal to psi s plus psi p. Water potential is a sum of solute potential and pressure potential. Okay, so water move from area of high water potential to low water potential. Normally, water move from area of high water potential to low water potential, right? As you can see over here in this particular vessel, a pure water is there. So water is present in the both the sides of the vessel, right? If and there is a membrane in between. If we put some amount of solute over here, then obviously water move from the area of high concentration. Over here there is no solute, so concentration of solvent, or we can say water, is more. So water moves from area of high concentration to low concentration. As over here solute is there, so water concentration is less, right? So it moves from area of high concentration to low concentration. And this movement down a gradient of free energy, this movement down a gradient of free energy called diffusion. Okay, now kinetic okay, just but if we allow pressure also on one direction, then also if we allow pressure on one direction, then also water move from the area of high pressure to area of low pressure. And if we so this is known as positive pressure. If we put up pressure on the water, if we release the pressure on this direction, that is known as negative pressure. That time water move from no pressure to negative pressure. Okay, now kinetic energy of water. Water molecule in liquid and gaseous form have rapid and random movement. Rapid, constant and random movement. Okay, so that is kinetic energy of water. So high water concentration is equal to high kinetic energy or high water potential. Okay, so we can say that high water concentration is equal to high kinetic energy or water potential. Okay, concentration of water is high when there is no solute dissolve in it. That type of molecule or pure water have high kinetic energy, and we can say pure water have high water potential. That's why the pure water has greatest water potential, that is psi w is 0 at standard temperature and if there is no pressure is applied. Okay, that time that particular water in which there is no solute, nothing, that is having highest kinetic energy in it, and that is symbolized as 0 at standard temperature and no pressure. Clear? Now let's discuss solute potential. Just like water potential, another term is solute potential. Means adding solute to water It decrease free water movement concentration. It decrease free water movement concentration. So it lowering the water potential. As just now we have discussed, pure water has highest water potential. If we add some kind of solute in it, like this is here, only water is there, right? So it is having highest water potential. But if we add some kind of solute like sodium over here, it hinder its free movement, right? It hinder its kinetic energy. So it decrease free water movement, or we can say kinetic movement of water. So it decrease its concentration, so lowering the water potential. Okay, so all the solutions have lower water potential than pure water. That's why in the previous slide we have discussed that pure water, in which there is no kind of solvent is dissolved, so not solvent, solute is dissolved, that is having highest water potential, that is 0. So obviously all solutions have lower water potential than pure water. Clear? So solute potential is always negative. As pure water potential of pure water is 0, then obviously if some kind of molecule we have dissolved in that water, that time the solute potential is less than this, so obviously in negative. Highest is this. So obviously if we compare this solute potential with water potential, and we just now we have discussed that all solution have lower water potential than pure water, then obviously the solute potential is must be present in negative. Clear? So the greater the solute concentration, the more negative the solute potential. This is again a very important line. Greater the solute potential, right? Or solute not potential, solute concentration. More the solute concentration in a solution, the more negative solute potential. Clear? Now, third term, which is also related with this topic, pressure potential. We have discussed water potential, we have discussed solute potential, now pressure potential means applying pressure to water. Applying pressure to water or a solution, it increase its water potential. If we apply a kind of pressure, okay, in a solution or water, it increase its water potential. So in plants, water entry into the cell create turgor pressure against the cell wall, which increase pressure potential. There is a cell, okay, there is a cell wall, cell membrane. So this water potential, okay, water entry into the cell create a turgor pressure against the cell wall. It increase the pressure potential. Clear? So pressure potential is usually positive, but it can be negative in xylem, water column, adding water transport up to the stem. Clear? In previous slide, just now we have, I just show you that slide also here. Pressure. If we apply pressure, just now in this particular slide, we have discussed this thing now. If we add some kind of solute, then water move from high concentration to low concentration from the this is known as water potential. If we apply a kind of pressure over here on the water, it will increase water potential, right? So movement of water occur from the high pressure to low pressure. This is known as pressure potential. Or if we release the pressure or we apply negative pressure, means release the pressure, that time also water movement occur from high to low. Low, why? Because we just release the normal pressure also. We have discussed this point also. Now let's move to the current slide, which just now we have discussed, pressure potential. Now, so I hope all three terms has been cleared: water potential, solute potential, and pressure potential. Now next term, that is osmosis. Osmosis means, osmosis is a type of diffusion. It is a type of diffusion of water molecule, or we can say solvent, across a semi-permeable membrane. This is important. So, movement of water across a semi-permeable membrane is known as osmosis. Okay. So, as there is a two section with the which is having semi-permeable membrane. So, here these are the solute molecule. This is water. Right. So, movement of water across a semi-permeable membrane, this is known as osmosis, and this occurs spontaneously due to pressure or concentration gradient. Okay. So, water move from the high chemical potential, or we can say high concentration, to low concentration until equilibrium is reached. Until equilibrium is reached. As you can see over here, concentration of water is more over here, as solute concentration is less. Concentration of water as compared to this A compartment is less over here. So water movement occur from here to here until equilibrium is reached. Means concentration in both the chambers A and B is equal in concentration. Till then, movement of water occur. Okay. So movement of molecule in plants, this one is determined by the cell membrane and the tonoplast, which is the outer covering of vacuole. Movement of water in plants is determined by the cell membrane and the tonoplast. And again, I'll repeat, this osmosis is a type of diffusion in which solvent or movement of water occur across a semi-permeable membrane until equilibrium is reached. Now let's discuss some experiments which demonstrate osmosis. First is potato osmometer, in which a potato tuber in water collects water in its cavity with a concentrated sugar solution. Okay. Another one is sucrose solution in a funnel. This is another experiment. Sucrose solution in a funnel separates from pure water by a semi-permeable membrane, or egg membrane is a type of semi-permeable membrane, shows water moving into the funnel, rising the solution until equilibrium. We can discuss this with this, or you can clearly I can clear you this experiment with the help of this diagram. This is the demonstration of osmosis. This is a funnel which is containing sucrose solution. Just now I have read it up for you. Okay. And when we put this funnel in inverted position in a jar which contain pure water, and this funnel mouth is covered with the semi-permeable membrane, then what will happen? Water move from high concentration, which is obviously present in the jar, to low concentration, means in sucrose solution. Water move from jar to funnel. Okay. So what will happen? The amount of sucrose solution is more from this level to this level. Okay. Water will diffuse. What is mentioned for A? Water will diffuse across the membrane to rise the level of solution in the funnel from this level to this level, as water move from high concentration to low concentration across the semi-permeable membrane. But if we apply pressure if we apply pressure like this on the funnel, it will stop water movement into the funnel. And this pressure is known as osmotic pressure. If we apply external pressure, okay, it will prevent entry of water inside the funnel, and this is known as osmotic pressure. Clear? So, osmotic pressure depends upon solute concentration. High solute concentration require more pressure. Low solute concentration just reverse. Right? Or vice versa. High solute concentration require more pressure means low solute concentration require less pressure. So, osmotic pressure, which is normally positive, osmotic pressure is numerically equivalent to osmotic potential, and this osmotic potential is negative. Okay, so this one is important line. Osmotic pressure is numerically equivalent to osmotic potential but with opposite sign. Clear? Now let's discuss different type of solutions. First is isotonic, then hypotonic, then hypertonic. Isotonic means concentration of a solute is equal in both the sides. Means external solution balance the osmotic pressure of the cytoplasm, in which if concentration is equal inside the cell or and outside the environment, then there is no net water movement occur. Okay, what is written over here? External solution balances the osmotic pressure of the cytoplasm, no net water movement occur. Clear? Hypotonic, external solution is more diluted. If there is a Petri plate and we have put it a kind of resin over here, okay, and this is just water, and this is a resin or kishmish in which sugar solution is there. Sugar. Then what will happen? External solution is more diluted, there is no solute, nothing. Then movement of water occur from high concentration to low concentration. Then after some time this resin or kishmish will swellen up as water is entered from the external solution to inside that particular resin. Right? Hypotonic, if external solution is more concentration, that time if this one is more concentration, here not water but sugar solution, highly concentrated, then what will happen? Then if we compare this resin, or if we put not resin, a plant leaf or something like this, if sugar solution is less in this particular kishmish or resin as compared to the outsider environment, then water movement is occur from inside the resin to outside. That time it will shrink as its water move from the resin and come out into the Petri plate or in solution. Okay, so there are three type of solutions. Isotonic, when osmotic pressure or concentration of water or solvent is equal inside the cell or in outside the cell, so no net movement of the water occur. Hypotonic, when external solution is more dilute, then water enter inside the cell, then time that resin swell up. Hypertonic, when external solution is more concentrated, that time just reverse to the hypotonic experiment, means the cells shrink it. Now another term is related to this topic is plasmolysis. Plasmolysis occur when a plant cell placed in hypertonic solution. Hypertonic solution means water move out the cell, causing cell membrane to shrink. cell membrane to shrink up from the cell wall. This is similar to hypertonic solution. Means when a plant cell is placed in a hypertonic solution, that time all the water which is present inside the cell, it just move out from the cell, and that time that leaf is shrink away from, and it will shrink, and the cell membrane shrink up and just keep the cell away from the cell wall. Initially water lose from the water which is present inside the plasma plant cell, it just lost from the cytoplasm and then from the vacuole also. Okay, and the protoplast means protoplast means all the content except cell wall. This protoplast shrink, and space is created between cell wall and shrunken protoplasm, as you can see over here. This is known as plasmolysis. If we put a plant cell in a hypertonic solution, then all the water which is present inside the plant cell is just move out from the cell, and then first from the cytoplasm and then from the vacuole also, and that plant cell gets shrunken up. Okay, now reversibility effect. This plasmolysis is usually reversible. If we put same cell, uh, if we put that particular plant cell in which is we have put it in the hypertonic, that shrinking, but if we just change the solution and now we just fill the solution with hypotonic solution, replace that particular hypertonic solution with the hypotonic, that time water enter inside the cell and causing cytoplasm to exert a turgor pressure against the cell wall in hypotonic solution. Okay, so this turgor pressure, what is this turgor pressure? It is a pressure potential exerted by the protoplast against the rigid cell wall. This is turgor pressure. It is a pressure potential exerted by the protoplasm against the rigid cell wall. Turgor pressure prevents cell rupture and is responsible for cell enlargement and extension growth. Okay, extension growth, as just now, just again, if we put this in hypotonic solution, this C, then what will happen? Water move from hypotonic, means concentration of water is more outside as compared to inside the cell, then water move from outside to inside the cell. Okay, and this time concentration of water, this is more inside the cell after some time, and it provide a pressure on the cell wall. So this pressure is known as turgor pressure or turgidity happen, and it is a pressure which is provided by the protoplasm against the cell wall. Clear? That's what it is written over here. Turgor pressure means it is the pressure potential exerted by the protoplasm against the rigid cell wall, and this turgor pressure prevents cell rupture and is responsible for cell enlargement and extension growth. Next term which is related to this topic is imbibition. Imbibition means diffusion of water. Diffusion of water, or water is absorbed by a solid or seeds which is present in a colloidal form, causing its significant increase in volume. This diffusion is known as imbibition. As this dry fruits, peanuts or badam, when we put it when we soak it in the water and put it as such overnight, and in the morning it will uh, imbibed water, right? So it will cause significant increase in the volume of this badam, right? So absorption of water by seeds is a type of imbibition. Absorption of water by dry wood also occur through imbibition, and this imbibition is essential for seedling to emerge from soil and establish themselves. Now, mechanism of imbibition. Water moves along a concentration gradient. Materials with low water content absorb water easily. So, all the materials which are having very less water in it, they actually absorb water easily through imbibition. It requires a water potential gradient between absorbent and liquid. It requires a water potential gradient between the absorbent and the liquid, and affinity between the absorbent and the liquid is necessary for imbibition. Okay. Now, so these are the methods for short distance transportation of water and nutrient. Now, let's discuss long distance transportation of water. So, long distance transportation within water can't rely on diffusion, because diffusion is a slow process. So, if we movement across a typical cell, which is 50 micron in size, through diffusion, it takes approximately 2.5 seconds. Over a meter diffusion, so for the movement of water molecule for a meter, this diffusion process would take years. But water is required every day for all the part of the plant and through the leaf, and similarly food is required by all the plant cells. If diffusion is utilized by the plant for the movement purpose, it requires too much time. So for long distance transportation, another mechanism is used up by the plant, that is vascular bundles through xylem and phloem. Okay, so movement of water, mineral and food in bulk due to pressure difference, substance in solution or suspended move together. Unlike in diffusion, bulk diffusion can occur via positive hydrostatic pressure gradient in garden hose and negative hydrostatic pressure gradient in suction through a straw. So translocation. Translocation means movement of water and food particle or nutrients in for the long distance. Okay, or we can say bulk movement of substance through plant vascular system, in which xylem and phloem is the main part. Xylem is used up for the movement of water and mineral salt and some organic nitrogen and hormones from root to other parts. So water movement occur only in one direction, while food movement in both the direction, two-way movement for phloem, or nutrient movement. Okay, so phloem transportation of variety of organic and inorganic solute, mainly from leaf to other parts of the plant. How plant absorb water? Initially, this roots specifically contains root hairs on the tip. Okay, these root hairs are actually primarily responsible for water absorption. Root hairs, in the anatomy of the plant, we have discussed the all the anatomy, means outer layer contains epidermis, and this epidermis extensions is known as root hairs, right? And after epidermis, cortex and other type of tissue is there. So root hairs are thin walled extension of root epidermal cell, which actually increase the surface area of this, and which is required for the absorption. Okay, so through root hairs, water is absorbed by the plant. Water along with minerals also, mineral solute, and this is from soil to root hair. This is absorbed by diffusion. Okay, and after that, once it comes inside the root hair, different pathway is followed up by the plant. First is apoplast movement, means movement through adjacent cell wall and intercellular space. Movement of water and other minerals occur through adjacent cell wall and intracellular space through apoplast movement, but it does not cross cell membrane. It depends upon the gradient. Okay, so mass flow occur due to adhesive or cohesive property of the water. Second movement is through symplast movement, means movement through interconnected protoplast and plasmodesmata. Movement of water through interconnected protoplast from one cell to another cell, right? Through protoplast and plasmodesmata occur via symplast movement. So within cell, okay, when there is no crossing between one cell to another cell, apoplast, but from movement of one cell to another cell through plasmodesmata or through interconnected protoplast like this, right? This is known as symplast. Okay, so if interconnected protoplast is there, plasmodesmata is there, so from epidermis to cortex to other parts through symplast, but movement of one cell to another cell. I'll correct myself. Previously, I told you within cell. No, not within cell, but from one cell to another cell, but through these channels within cell membrane, within cell membrane. Okay, via this apoplast. As this apoplast means movement through adjacent cell wall and intracellular space. And this apoplast movement of water does not cross the cell membrane. Does not cross the cell membrane. So movement, it doesn't cross the cell membrane within intracellular space. Okay, movement of water occur through apoplast, and in symplast movement of water occur through protoplast and through plasmodesmata. Okay, water enter through membranes and making the process slower, which is aided by cytoplasmic streaming too. Okay, so once it cross root hairs wala part, then cortical cell and endodermis also come inside the we have already discussed the cross section, right? So water move from roots via apoplast due to loosely packed cortical cell, and then endodermis. As you know, this is having suberized Casparian strip, and this endodermis is impermeable or impervious to water. So water is further redirect to non-suberized region and move through symplast to reach the xylem. And this is important point. Sometime question ask on this particular line. Water doesn't cross the endodermis. Rather, water is redirect to other cells where it used a symplastic movement to reach the xylem. Once it comes into the xylem, it will freely move uh, through xylem vessels and tracheids, which is a part of apoplast. Okay, so water and minerals move freely between and through the cells once it comes inside the xylem. Now, additional structures are also there, like mycorrhiza. It's a very common term, and it's a very important term also. So many examinations, so many question asked related to this particular topic, mycorrhiza. Mycorrhiza is a symbiotic association between fungus and um, some plant root system. Okay, this is the fungal filament. This fungal filament increase the surface area for the absorption of water and nutrient or minerals, or not nutrient, minerals. So fungal filament increase the surface area for the absorption of water and nutrient, while so it will help the plant to absorb more water, more minerals from the soil. Okay, and roots of the plant, mainly pinus, roots of the plant provide food or nutrient to the fungus. So it is a plus plus relationship, or in which both the partners get benefit. Okay, fungus is benefited by the plant, as it is provided, this plant provides sugar and nitrogen compound to the fungus, and fungus provide water and minerals to the plant. Okay, so this symbiotic relationship is known as mycorrhiza. It is a very important example of symbiotic relationship which is present among fungus and pinus roots. So this is all about the today's video lecture. I hope most of the things has been cleared now. If still you have any query, any doubt, please let me know. We will discuss it later on. Till then, keep learning and keep um, reading all the topics related to this particular chapter. Namaskar.